Hello, everyone. I am Verena Ras. I chair the Sample Collection and Processing Subcommittee within the African BioGenome Project. Today, I will tell you a little more about our experiences with the African Genome Project and sample collections, our challenges, our solutions to some of those challenges, and also some of our successes over the last month or two. So just to tell you a little bit more about the African Genome Project, firstly, for those of you who might not be too aware of the African Genome Project, the African Genome Project is really a project that's aiming to sequence every endemic species on the continent. So it is a monumental effort. But essentially, Africa BP has three main goals. The first goal and overarching aim is to coordinate all non-human genome sequencing on the continent. All of these initiatives, we aim to coordinate them and to act as a forum, especially for the non-human space. We also want to initiate, develop, and support new non-human genome sequencing efforts with the goal of sequencing in excess of 105,000 species across Africa. And we also aim to deploy sequencing data to contribute to regional data banks, biobanks, and to also, of course, inform policy and build capacity across Africa. So Africa BP aims to do this via four key programs. Our first program is our Animal Genomes Program. Our second program is our Plant Genomes Program. Our third program is our Community of Practice on Biodiversity Framework. And Program 4 is our Open Institute for Genomics and Bioinformatics. And Africa BP's overarching role within all of these programs is that of a coordination role. We will be focusing on fundraising, partnerships building, and policy engagement and deployment. So this is a bit of a timeline of the African BioGenome Project. We are currently in our pilot trial phase. With this phase, we aim to sequence up to 2,500 endemic African species. And of course, we do aim to distribute these species sequencing efforts across regional diversity and taxa. And value proposition is, of course, very, very important to us. Once we complete this pilot trial phase, we'll move on to phase 1, which is a bit of a large scale phase where we'll be doing the predominant sort of, or the most of our sequencing. And then with phase 2, we will basically reassess what we've done with phase 1, implement some recommendations, and also sequence any additional taxa that we might not have captured with phase 1. But just to come back to sample collection and processing, which is the committee that I lead, the Sample Collection and Processing Subcommittee forms part of the African BioGenome Pilot Project, as I've just mentioned. And so our main role within the pilot project is quite vast, and it expands or spans quite a lot of sort of areas and domains. So we manage quite a lot of things. So some of the things that we are responsible for have been placed on the slide. So we try to, of course, ensure adherence to legal and ethical frameworks across the continent. We are responsible for curating species and the metadata. So this is also biospecimens. So we do work closely with biorepositories to also store some biospecimens. We are aiming to provide quite a lot of collection and processing standards, and I'll speak to that in just a second. We also are responsible for coordinating the national representatives across Africa. So national representatives within the context of Africa BP are national access, benefit, and sharing focal persons that are situated in various countries. And our aim is to have at least one ABS focal person per African country who can assist us with adherence to legal and ethical frameworks, policy issues, and so on. We also manage taxonomic working groups. Our taxonomic working groups have been formed to help us prioritize our species lists, our species coming in through our nominations, and also to just ensure that our species IDs and so on are accurate, and we are using accurate sort of frameworks and mechanisms to also do our IDs and so on. But then, of course, we also play a major role in sourcing species and then liaising with our sample ambassadors, who are our sample custodians, but we call them ambassadors with the Africa BP project. So as I said, we aim to sequence with the pilot project up to 2,500 endemic species. These may include endangered species, and we are aiming to include some endangered species in that. But you can imagine that that would present quite a number of challenges. But I think it also puts us in a prime sort of position to come up with some very unique African solutions. And so I think Africa BP will really play a major role in finding some of these solutions and harmonizing a lot of the efforts across the continent. So we do aim to engage with a lot of sample custodians in different domains. Our main aim, of course, is to collect fresh material, but as we know, within Africa, this might not always be readily available. And so we have embarked on some initiatives to also engage with, conservation centers, aquariums, zoos, to try and collect some tissue that way. But we have also engaged with some museums. And I'm going to talk a little bit about some of our accesses with museum samples in just a second. But of course, very important to us is that local taxonomist communities and even indigenous communities are engaged about anything that we do. And any contributions from even indigenous communities will be recorded as we collect our samples. And so that is very important to us as well. So this is a bit of our snapshot, really, of our sample collection and processing sort of pipeline. It is not all encompassing of all the things that we do, but it's really just a snapshot from when we decide that we're going to collect the sample from you, all the way to when we've assembled the sample and we've stored it in the cloud. So I won't speak to this in any great detail, but you can just see the major steps on this slide. So it will always start by, of course, us confirming your sample submission, assigning some type of ID to your sample, and you collecting the tissue. That's always your starting point. Then we've got a range of documentation that needs to be completed. Again, I'm going to touch on that in just a second, after which your samples are prepped for shipping. It's shipped to our labs, it's sequenced, it's analyzed, and eventually the assembled genome will hopefully be submitted to a central repository for everybody to benefit from or access. And this is a process that will likely change a little bit as we progress with the project and as we learn, but at the moment, this is the process that we are going with. So in order to assist sample custodians across Africa, we have developed terms of reference as a sort of a guide for our sample custodians, which contains quite a lot of information that is quite sort of pertinent to the collection of samples. So we have our considerations, for example, for selecting representative species, so that you know immediately what species we're targeting upfront. These are obviously the species we're prioritizing at the moment, but again, this might change as we move along. But the terms of reference also provides a whole suite of information about ethical collecting instructions, metadata collection, and we have a whole metadata template that we've set up that does link to other initiatives. So it does link to sort of the Darwin Tree of Life and what sort of metadata fields that they suggest we should be collecting, but we do obviously also collect some Africa BP specific metadata as well. But it also contains things like how to do your specimen and tissue shipping, some best practices, some information about data ownership and IP. So it's a really, really nice guide in essence for those who want to contribute to the Africa BP project. But I must warn you that our LC team, that is our ethics, legal, and social issues team, have come up with a range of documentation that every sample submitter must have in place for us to be able to accept that sample and sequence it. So these are permits, MTAs or MATs, so mutually agreed terms, prior informed consent. There are also letters that we require from your country, consenting, sample shipment and so on. So we have to work via national and regional laws. Obviously, we have to comply with that, and that becomes very tough sometimes. And I will address that in just a couple of minutes. But here are a couple of our key achievements. I'm not going to speak to this at any great length because I believe you will receive a detailed talk about some of the sequencing efforts that are underway and have been completed. But I thought I would highlight a couple of key achievements, which is one of our sequenced genomes. This is the very first genome we actually sequenced under the umbrella of Africa BP. So this was a Rhinotyphlops lelandii. So that's a big blind snake. And you can see from just the snapshots I've got here that the sample quality was really excellent. The sample concentrations, very, very excellent. And the total sort of DNA that could be obtained was quite good. And you can also just see from these peaks that actually the sample was really, really good. And so that this excited us because this was actually from a collection. I'm not too clear, but the sample was actually, I think, more than 2 years old and sitting in the collection, and we're still able to actually obtain really high quality DNA. So this was very exciting for us because it showed a lot of promise for adapting sort of techniques within Africa to make it work within an African context. And this effort was in collaboration with the Vertebrate Genome Project. SANBI, as well as HA BioNet and UNISO, contributed to this quite a lot. Again, there will be a talk that will speak to some of these results in a bit more detail, but just for those who might be interested, here are some of the output metrics from the sequencing assembly. So you can see some of the assembly metrics here. I won't speak to that at any great depth, but in case you're interested, you can see some of the N50 values, for example, our number of contigs, the total length. It's very, very good, actually. And we actually covered, I think, more than 90% of the genome. So that was great. We had really high coverage. And I also think with this particular sample, we had really good sequencing depth. I think we had in excess of 30 times, which I was very excited about. And that is in terms of an achievement for our animal genomes project. A key achievement for our plant genome sort of program or project is that we have embarked on sequencing a helichrysum, which is a culturally important species within South Africa, especially. But we have also sort of started to sequence other species, which I've kind of listed on the slide. So we are starting to sequence and push out some data. So that's very, very exciting. So I don't have species names here, just some common names. But these are some of the current/recently sequenced species. So we've got things like endemic Nguni cattle, Nguni sheep, Nguni goat, which are endemic to South Africa. We've got the bush tea, which we are very excited to see, and we hope that that works and it comes out really well. But also the Goliath heron we've been working on, and the West African mud turtle, but hopefully there'll be a lot more on the way very, very soon. And so we've used this, of course, to start to put a lot of our policies and so on in place. And in doing so, we've also held these sort of community of practice roundtable meetings where we invite ABS focal people to share with us, to inform us, to help us, to guide us. And this has been very well attended and very exciting to see ABS, focal people coming on board and teaching us, how to do things better. We have also run a couple of workshops now where we're also trying to disseminate what we learned to the broader community. Not going to say too much on this now, but happy to touch on this when we have a bit of Q&A. So what are some of the challenges that we've experienced as the African BioGenome Project? There are a few. Again, not going to go into any great length, but I'd just like to mention a few of them here. Of course, funding for sample collectors is difficult. A lot of sample collectors struggle with sourcing funding, permitting costs. There's an unequal landscape in Africa, which is very difficult to navigate, storage and preservation, shipping issues and costs. So there's lots of challenges, but we have also identified a few solutions. Some of which have been listed on the slide. So these are obviously things like obtaining funding, having ABS discussions early and entering into bilateral agreements, countrywide collection permits, and practical guidelines and training for how to do this in Africa. These are just some of the solutions. These are obviously not everything, but I hope that I've given you a very good summary of what we've been up to as the Sample Collection and Processing Subcommittee. And so these are just some of the members I'd like to thank who have contributed quite a lot to the African BioGenome Project. Thank you very much.